Thanks, Dr. Voda, for that kind introduction, and also thanks to Dr. Conley and Chelsea for setting this up so nicely, because we're going to really build on what we've been covering before the last two talks. Let's see if I can get this to go. All right, so we're going to talk about research updates in limb-girdle muscular dystrophy. So that's a very sensitive clicker. So these are my disclosures. We'll be talking about. I've gotten slides from a number of the different pharmaceutical companies in this disease, and so I'll try and point those out. So I divided my talk into two different discussions. So the first is really preparing ourselves for the upcoming clinical trials, and the second is talking about the different types of treatment approaches that people are thinking about across the different limb girdles. And so I always think about these, developing new drugs and getting these through the process and approved for use in our clinic is a very challenging thing, and it really has to be full partnership between all of us, both the researchers, the patients, and the pharmaceutical industry to get this to the right place. And so, we'll talk about kind of the different steps and where we have to meet. And really, it only works if it meets as a perfect intersection. So, talking about clinical trial preparedness first. So why now? Why are we are thinking about this now? Why are we talking about this today? So our ability to diagnose limb-girdle muscular dystrophy has improved, as Chelsea elaborated on. And so this is an example. This is a slide showing the results of that free genetic testing program that the MDA and Jane Foundation sponsored. So you can see that for the first time ever, we've got a lot of people who were sitting and didn't have a good diagnosis and now have a definitive genetic testing result, as Chelsea alluded to. And that's really because of the advocacy programs that have sponsored these free genetic testing programs, and the cost of commercial testing is lowered. So that's a win. That's exciting, and that we're able to understand these diseases a lot better if we can with good genetic diagnoses. But one of the problems is that there's a lot of what that uncovers, that there's an increasing number of individuals with variants of unknown significance or negative testing. This is, again, this is really building off of what Chelsea was talking about. At the same time, advances in gene therapy have identified limb girdles as really key targets for gene replacement therapy, and I'm going to talk about that in the second half. So that's a very exciting development. And researchers and companies are developing other molecularly targeted therapies. And so I think there's a lot of hope and promise being developed on the laboratory side. So there's a real possibility that several clinical trials in limb-girdle happening over the next 5 years. However, our problem is that I think Dr. Connolly very much emphasized this. This came up again in Chelsea's talk. These are incredibly variable diseases, and so in order to do a good clinical trial, you need to be able to understand and say, well, this is what we think would happen normally if we didn't treat the condition. And so we're way behind. We're the tortoise here in terms of trial preparedness, and this is a happy problem. This is a good thing to have, that we have these new and exciting therapies, but we need to start to kind of catch up with where the science is in terms of gene replacement therapies. So we've identified as a research community that there are really several gaps that we need to address. Gap number one is how we diagnose people with limb-girdle muscular dystrophy. Gap number two is the tools that we use to measure response to therapies as part of a trial. And gap number three is in our understanding of the patient experience of living with limb-girdle muscular dystrophy. It's all well and good if we can show on a muscle biopsy that we can restore the protein, but it doesn't make a whit of difference if it doesn't make you feel any better, or allow you to do what you're interested in doing. So we formed a clinical trial research network, which can be defined on its simplest level as a group of academic or private research centers who agree to work together to accomplish a shared set of goals. And it creates a more efficient process for us to run clinical studies, and there's power in numbers. So we can implement rational diagnostic workflows and reach more people, and because we've all come together and agreed to pool all of our resources for the shared set of goals, we can actually answer these trials or answer some of these questions more effectively. And we can ensure that the findings, it's all well and good if we know how to do things well in Richmond, but it doesn't do, again, any good if you can't replicate it in California. And so, we've sought to include multiple sites across the country and the world. Findings are more likely to represent the broader LGMD community. So this is the consortium that's been found. So it's the Genetic Resolution and Assessment Solving Phenotypes in Limb-Girdle, so the GRASP LGMD Consortium. And so right now this has been supported by the Muscular Dystrophy Association and the Coalition to Cure Calpainopathy. And you can see on the map here where the sites are. On my very last slide, we'll list where the different sites are. And the goal is really, like I said, to hasten therapeutic development for limb-girdle by improving the diagnostics, developing outcome measures, understanding patient-reported disease impact, and to develop new biomarkers, or the ability to measure how the disease progresses. And of course, we're very interested in making sure that other people enter the field as researchers in limb-girdle. So, talking about specifically GAP1, because of the possibility for gene replacement therapies and to help guide medical surveillance, one of the projects that is beginning is looking to understand those variants of unknown significance. And so everyone who presents with weakness in hip or shoulder girdle in which their doctor feels is due to an inherited muscle will need genetic testing. About 20 to 25% will end up with a confirmed diagnosis if we use the currently available panels, and about 55% will have one of these variants of unknown significance, which is a significant gap, and about 20 to 25% will have no mutation. So what's next? So we are working to resolving these variants. So we've done a very good job at identifying them, but we're working with colleagues at Washington University and Yale to help identify those variants and resolve them. And then here are some of the ways that we think about doing that. So one is, does that variant track with affected individuals in your family? So I think Chelsea alluded to that a little bit. Or in large groups. So again, since we're collecting variants from all over the country, we can start to see if they keep showing up over and over again, is there a way for us to see if it occurs in many of the affected individuals? That's one of the ways to resolve that uncertainty. And then you can see here, Dr. Conley already gave you your pathology talk early. So you can see here, this is again the same slide. And so you can actually stain for a lot of these muscle membranes. Here, this is an alpha-sarcoglycan, same example that Dr. Conley used. And here, in this patient with limb-girdle 2D, you can see that you don't see that muscle stains appropriately. So that's a good way to know whether or not that variant actually, so that uncertain genetic testing result actually, this is a way to provide certainty to it. And there's other ways to look at it as well. Again, looking at functional assays or other techniques looking at messenger RNA. So the workflow, right now, this is currently being done by a number of different groups, and so everybody's doing something slightly different, and what we've agreed to do is to standardize that workflow and to be adaptable to each person's mutation. So this should be coming down the road, and will certainly provide a better diagnostic process for people in the community. In addition to that, what happens if there's no mutation? So some of our colleagues at Yale University are looking at expanding what they can test. So right now they currently test for a short list of genes. So looking at either whole exome sequencing or whole genome sequencing to looking at genes that are not normally tested. And then looking at other techniques such as RNA sequencing, which is a more specific way in the muscles to look and see whether or not you can find a gene that's causing that muscle weakness. And so, again, pooling our resources, coming together as a community, and looking at a larger group of people allows us to identify these new genes. As part of addressing that gap, really our goal is to enroll up to 2,000 people with either a variant or no mutation, and to create a rational workflow to resolve those variants of unknown significance, and to start to find other genes. So I know certainly this came up multiple times, but we're on number 34, and there's certainly a chance that we're going to find number 35 or number 36 in terms of genes that can cause limb-girdle muscular dystrophy. And then, we've been working closely with partners to continue to offer free genetic testing. So gap number 2 is really the tools for clinical trials. So these are outcome measures, are tools that we use to tell if something in limb-girdle has changed between 2 points in time. So there's lots of different outcome measures that people talk about this all the time. So they can be measured in the blood or in the muscle, in which case they're called biomarkers. They can be something that a physical therapist or another evaluator assesses, like strength or the ability to do tasks, or they can be questionnaires that you do, that you talk about your experience with limb-girdle muscular dystrophy. So we focused, what are good outcome measures? So these are things that are pertinent to the disease, that are reliable. So if you measure them one day or the next, they look the same. Very importantly, we're very interested, because oftentimes limb-girdle muscular dystrophy is slowly progressive, we're interested in finding things that detect change slowly, or that are sensitive to slow change, because that's going to be really important. That allows us to do shorter clinical trials. That are inexpensive and, of course, feasible to collect at a number of different sites. So these are all the things that we're thinking about when we think about what makes a good tool to use in a clinical trial. And so it has to be the right tool for the right job. Measuring gene expression, for example, is not going to tell you whether or not somebody's experiencing pain. So I think that's an important point. And that different tools are important at different stages of the drug development process. So lots of times early clinical trials focus on getting that muscle biopsy and showing that they can return the protein expression. It's much more important in later phases to show that that's meaningful to you. So whether or not the protein is expressed or not expressed, that only tells us, again, like I said, that the protein's expressed, but it does not tell us whether or not you can walk better. So you really have to capture the right thing, and so we focused on building a clinical trial toolbox to have the full armamentarium available. And so for biomarkers, just a word about that. So for gene therapy, the obvious biomarker is expression of the missing protein, which is similar to what they use in Duchenne muscular dystrophy. You've seen a lot of pictures now of muscle, and so everyone here is an expert. And you're going to see more of these as I talk about the different gene therapy programs. So this is now a fluorescent image. So it's the same muscle membrane. It's still cut in cross-section, so you're still looking at it kind of on its side. And here what you're doing is you're staining for a very specific protein. And there's lots of different proteins you can stain for. So you can stain for the sarcoglycans or the dysferlins or the calpains. There's lots of different proteins you can stain for. It's not going to show up very well in the background, sorry. But here you can see that there's the staining. This is a control muscle, and this is somebody who's been treated, and so you can see that the protein's been returned. But for some limb-girdles, there are other things that we are interested in measuring in the blood. So evidence of muscle destruction, the one that we commonly are thinking about, depending again on the type of limb-girdle, is a measurement called the CK, which is pretty simple to use, and in some limb-girdles, not all, it can be quite elevated. And so it's a good measure of muscle destruction and injury, looking at other inflammatory markers. Another biomarker that our group is starting to explore is muscle MRI. So this is again, I'm sorry for the size and the background, but this is a group of muscles that were looked at, and these are the patients that they were looked at. And here in the green, you can see that's a signal that's saying that muscle doesn't look right. That looks like it's been damaged. And this, again, is a thigh here. And so you can see that this muscle here looks quite normal. They're supposed to look kind of black. and that as muscles get damaged, they end up with a little bit more of the kind of light signal in there. So that's a good way of measuring change. But there's a lot of work that we're doing in our group, again, trying to prepare for clinical trials. So we need to ensure that these biomarkers are reliable, that they respond to treatment, and and that we understand what a change in biomarker is in terms of how it would affect clinical function. Strength and function, this is, of course, my favorite thing to look at, and this is, probably the key anchor for any type of clinical trial. So again, all of the limb girdles affect your ability to do this muscle sprint, or sprint across the front of the room here. And so we need to find ways to measure that change over time and do it in a sensitive and reliable way. So we do have lots of different ways that we use to measure strength, so manual muscle testing, something called quantitative myometry here, which is this kind of jungle gym looking tool, and then dynamic strength here. So there's lots of different ways to measure strength. Strength is only one part of it, so you can be strong in some muscles but not others and still not be able to walk well. And so we're interested in looking at how you walk, how you get up from a chair, how you go up for stairs and how far you reach. And we've been taking advantage with our colleagues at Nationwide Children's of using current technology to better capture this. So this is a tool that was developed by one of Dr. Conley's colleagues, Dr. Lowe's, which is actually, this is a boy playing a video game, but what it's really doing is actually measuring how well those limbs move. So, trying to take advantage of what people are interested in doing. And then, of course, we're looking at ways to combine those into a composite score. Again, lots of work that needs to be done. So we need to standardize our equipment and procedures. We need to train our evaluators and ensure that the relationship between genetics and age and gender and functional status are all understood as we embark on these upcoming clinical trials. And so this is just my brief advertisement, which is that Brittany and Sarah and Liz are outside, and so if you are interested or think you might be interested in doing one of these, they can certainly jot down your information, and there's a whole team of people, and so they can get you to a site that's closest to you if you're interested in that study. The last thing is trying to understand the patient perspective. So the choice of what you measure needs to match your perception of limb-girdle muscular dystrophy. So, for example, cognition or thinking is not really affected in limb-girdle muscular dystrophy. So, if we focus all of our tools on measuring IQ or how you're thinking, you're going to say, well, that doesn't matter anything at all. That's not what the issue is. And so, we need to make sure that the tools that we develop match your perception of what the issue is in limb-girdle muscular dystrophy. So we're in the process of developing a limb-girdle specific health inventory, which is a questionnaire that can be used in clinical trials. And so we started with 20 patient interviews and themes, of course, that came out in terms of mobility and ambulation, but other things like respiratory issues or cardiac issues may be important to some subtypes of limb-girdle. And so the next step is a survey that will be going out to talk about each symptom and how impactful that is on your life. This is just a graph showing the results of the different quotes that those 20 patients responded. So it's not surprising that most of the issues had to do with mobility and ambulation, inability to do activities, social role limitations, emotional distress. And so when we're developing our clinical trials and outcome measures, we are very much focusing on capturing things that affect mobility and ambulation. So, we're trying to listen to you. This is a partnership in making sure that we capture the things that are important to you. And then the last piece in terms of getting ready for clinical trials is really trying to understand disease progression. I really liked Dr. Conley's early point about that we're all in this together and that you should pay attention to your neighbor. So this is a graph here of some boys with Duchenne muscular dystrophy. And this was work that was done in Duchenne now a while ago, about 5 or 6 years ago, that really informed how they designed their clinical trials. And what you can see here is that this is the boy's age here, and this is how well they're walking on a 6-minute walk, and this is each individual line is a single boy, and you can see clearly that about age 7 that the walking really starts to decline, and that was very helpful in understanding that is a time when the disease really progresses, and that's an appropriate age to start to develop early clinical trials. So we don't have that type of information right now in limb-girdle. The second point of that is that if you tested the drug early here and used a 6-minute walk, these boys are actually improving. And so if they were in your placebo group, you would reject that therapy, even if it was actually working. And so, it's important that we develop this information. Of course, that's what we're trying to do with our consortium. We do, of course, these early clinical trials are testing new therapies, and so they do contain risk associated with them. And so we're interested in making sure that as few as people need to be exposed to that risk as possible. So it's a more efficient clinical trial. And to reduce the burden on people who are participating, there's a lot of work and effort that goes into participating in a clinical trial. And so we want to make sure that is, again, as efficient as possible. And to design those trials as effectively. So there are still several gaps in our understanding. So how do genetics, baseline functional status, demographics influence the rate of progression? And are there simple tools that we can use to predict how somebody might do over the next year? So again, these are all questions for us and for you as the research community as we get ready to embark on these next phase of clinical trials. And that's really emphasizing the point and the reason why we do these natural history studies. That's what we're here for. One brief side note before I embark on all the preclinical therapy development is that beyond these natural history studies, there are registries. I think you're going to hear more about the MDA Mover Data Hub, which will soon include limb-girdle muscular dystrophy. And it's just another way of collecting some of that clinical natural history data that really helps to inform the design of those clinical trials. So the summary from this half of it is that clinical trial preparedness is a collaboration between the industry, between academics like myself, basic science advocacy, and you, people with limb-girdle. Understanding the natural history and how we measure things helps us gain approval for new drugs. It ensures that if a drug is actually working, we're not going to say that it's not working if we design it effectively. And having a network gets sites ready to participate in those clinical trials. It creates a national group of trained coordinators and evaluators and helps individual investigators take their drugs into clinical trials. So there are many ways to participate. You can directly contact the project manager, which is Brittany Holmberg, and she'll get you to the right place. You can find the trials on clinicaltrials.gov, and there's a website that's under construction. And so this is really trying to work on a collaborative approach to making sure that we can come together and test these new therapies. Okay, so moving on to the therapies in development. I apologize in advance. There's a lot of technical detail on these slides. I'm just going to try to hit some of the high-level stuff, and you're going to see a lot of pictures of mouse muscles. So I'm going to apologize in advance. But I did try and break this down into 4 categories, because when we start to think about how to develop drugs to treat limb-girdle muscular dystrophy, we think about them in 4 large buckets. So the first is the gene replacement therapy. The second is small molecules. The third is something called antisense oligonucleotides, and the fourth is gene editing. And so I'm going to go through each one of these in detail to kind of tell you what I'm talking about. So gene replacement therapy, this is among the most advanced technologies. And so this figure over here shows what it is. And so basically, what you're doing with gene replacement therapy is replacing a defective gene. And all of the current approaches use the same basic idea, which is that you have a virus that is not considered to be pathogenic to humans and typically you haven't been exposed to, so there's not a presumed immune reaction. That the bad genes inside the virus are taken out of that, and the gene that's missing, so calpain-3 or the sarcoglycans or dysferlin, etc., is put into the virus, and the virus is then put into the person. So that's how it works, generally speaking. The virus, the gene that's put into the virus never fully becomes part of your genome. So it stays outside. So I have, how many people are familiar with chickenpox? So you've had chickenpox as a kid, and you never really lose that virus. And then if you're older, sometimes you'll get shingles, which is thought to be a re-expression of that viral protein. So the virus is not with you or stays with you for the rest of your life, but it doesn't become part of your genome. So in other words, you wouldn't necessarily pass it on to your kids. So that's just an important point about gene replacement therapy. So that's basically the way that it works. This works best in autosomal recessive types of limb-girdle. So you're taking, in autosomal recessive types of limb-girdle, oftentimes you're missing a gene, and so you can replace it. In dominant forms of limb-girdle, oftentimes you have an overexpression or a toxic protein that's in there, and so you need to think about a different strategy. Not to say it's not possible, but most of the gene therapy work is being done in the autosomal recessive types of limb-girdle muscular dystrophy. These are likely one-time therapies, and they do carry a potential risk of immune reaction. So this is, again, everyone, like I said, is now very familiar with the dystrophin protein complex. This is just yet another example of it. So basically, again, showing that you have your muscle membrane here, that you have the sarcoglycans here, and these are attached inside to the dystrophin protein and a number of other proteins that cause some form of limb-girdle muscular dystrophy. I really like Dr. Conley's sailboat analogy, so her slides were better than mine on this particular point. And so these are just again showing the different proteins, and to reorient you. I'm going to show you some examples of mice that have been treated. So these slides were provided by Sarepta kindly. And I'm sorry that my mouse over there is not working quite as well, so I keep leaning this way. But I'll show this on this side first, that basically this is a mouse that is deficient or has a limb-girdle 2E, essentially. That's the easiest way of thinking about it. And so basically they've used this AAV vector, including the sarcoglycan beta gene. So LGMD 2E is caused by a loss of sarcoglycan, the beta sarcoglycan. And you can see here that on this side, that these mice, you can see that they don't express the sarcoglycan, and then if you give them the gene therapy, that you can see that you can restore the expression of the sarcoglycan protein. So that's quite promising. This is again showing, again, it's important to show that you can restore the protein, but what does it actually mean? So here you have the sarcoglycan knockout mouse, and you can measure their strength here, and so you can show that once you restore once you provide the gene therapy, that they have an improved strength in the mice, and that their endurance is better. So again, this is just in a mouse showing that the mice get stronger and that they have better endurance. Here's a different example now provided by our colleagues at AskBio, looking at LGMD2I, which is FKRP. And I'm going to switch sides again here. So again, another muscle membrane stain here for a mouse model with essentially limb-girdle 2I, and again using a gene therapy vector to provide that FKRP gene back in, and you can see that the mouse has better restored protein expression, and that if you again looking at their CK. So a measure of muscle enzyme, that their CK comes down over time. And if you look at their endurance here on a treadmill, that they have an improved strength. So again, another promising result from a mouse model with LGMD2I. So this list shows all of the gene therapy programs that I'm aware of that are in existence or have been announced in some iteration. So this list of genes and subtypes are the genes that are kind of the first up in terms of the gene therapy programs that will be approaching clinical trials. Moving on to small molecule approaches. The way small molecules are, these are all the drugs that you have in your kitchen cabinet or in your bathroom. So these are aspirin and Tylenol, and 99.9% of all drugs are small molecules, is a way of thinking about it. So in drug development parlance, they're called small molecules. These are typically tend to be easy to deliver. As opposed to the gene replacement therapy, which is pretty self-explanatory, that you have a gene that's lost and you replace it. A lot of the small molecule designs try and work something downstream. So they try and find a protein that's upregulated protein that's misregulated because the protein's missing, or some other problem around that. I'll show you a couple of different examples of that. And these are much more traditional in terms of their trial pathway and approval pathway from the gene replacement therapies. So this is a small molecule potential for LGMD2I, which is FKRP again. So this is showing here that what you have with the mutant FKRP, normally you have this ribitol that's supposed to end up adding extra sugars to it. And because you lose that mutant FKRP, it doesn't get made appropriately on the outside of that muscle membrane. So essentially, what this trial is trying to do is to add extra ribitol, so it just over delivers more and more of the sugar that is blocked. And when it does that, it can actually kind of override the problem with that mutant FKRP and restore some of the sugars on the outside of the muscle membrane. Is an easy way of explaining it. And this is I'm very sorry in the back, but this is again another muscle, with another mouse with FKRP, and you can see here that the mouse on this side has all of the same bad problems with their muscle that Dr. Conley showed, and if you give that mouse the ribitol therapy, the muscle pathology looks better. And they have less scarring around the muscles. And then again, you start to get a theme that basically all of these studies are doing the same thing. They're looking at the muscle pathology, and then here this is an example where you're looking at the strength and endurance of those mice with LGMD2I, and they have improved strength and endurance after you give them the ribitol therapy. So again, another promising treatment for LGMD2I. Not to mistake our colleagues with dominant forms of limb-girdle muscular dystrophy. So this is a slide that was given to me by a colleague within the consortium, Dr. Weil at Washington University, who's been working with this drug called BGP15. It's not worth going into too much of the detail about it and what causes the problem in DNA JB6 or LGMD 1D mutations, but essentially what he's doing is trying to override that mutation and allow the proteins to function normally. And here he's looking at the number of cells with these aggregates, these toxic protein aggregates that get formed in limb-girdle muscular dystrophy. And when he treats them with this BGP15, he's able to show that the protein aggregates go down at the cells function in a healthier manner. So another small molecule approach that's still very much being worked on at the cell or the mouse level. And I think one of the things that you've now taken away is that we've very effectively treated a lot of mice. So I'm going to just cover, because these next two ideas, these next two therapeutic approaches have come to fruition in other forms of muscular dystrophy, or being tested in other forms of muscular dystrophy or neuromuscular conditions, and have not really been used a lot in limb-girdle muscular dystrophy. But I thought I wouldn't be complete if I didn't mention them as what people think about when they think about drug development. So this is a technology called Antisense Oligonucleotides. This is essentially what you're doing here is now you have your DNA, which is your that's your sentence structure that both Dr. Conley and Chelsea did a nice job of explaining. It then becomes a RNA, and what an antisense oligonucleotide is an artificial piece of RNA that binds to it. And when it binds to it, it can make it do one of two things. So it can either be used to change how the sentence reads to make it read the right way. So the cat in the hat analogy, and that technology has been used in Duchenne and spinal muscular atrophy. It can also be used to knock down gene expression. So as I kind of alluded to early on, so some of the dominant forms of limb-girdle where you have a toxic overexpression, this is a type of strategy that in the future people may think about as they get more familiar with the idea of the antisense oligonucleotides. And it hasn't. The challenge is that it's very hard to get artificial pieces of RNA into the muscle. And so there's lots of people, lots of basic science researchers who are trying to figure out how to get it into the muscle better. And that as that advances, I think we'll see more and more strategies trying to use this type of technology. So it hasn't really had a lot of use in limb-girdle muscular dystrophy. And last but not least, everyone's favorite, Dateline or CB 60 Minutes of the CRISPR technology. So I'm just going to cover that very briefly. So essentially what CRISPR is, it's called CRISPR-Cas9, and here what you're doing is that, it's probably best shown in this figure, which you can't see very well in the back, I apologize, is basically you use this CRISPR technology to cut out the bad parts of the gene, and you can either insert in a new gene or new good parts of the gene, or you can just basically cut out the gene and make it go away. So you can certainly, it's a gene editing therapy. So as I told you before, that the gene replacement strategies, this works like a virus, not like shingles or chickenpox, where the virus stays in your cell, but it never becomes part of your genome, your human, your fabric. CRISPR therapy actually goes in and edits your genome. And so this has a lot of promise and hope associated with it. But as you might expect, when you're starting to edit somebody's genome, this is like your master code. There's no backup for it. You want to make sure that you're not editing the wrong parts of the genome. So it's been used. There's lots of, like I said, hope and promise. It's been shown to work very effectively in mice with Duchenne muscular dystrophy. There are certainly people who are starting to think about using this for human clinical trials, but there's this large safety concern about making sure that it's precise and you're not editing the parts of the genome that you wouldn't necessarily want to edit. And so I think that this is not it needs to be evaluated preclinically, and certainly is not ready for the first batch of therapies and clinical trials that we're going to see. So I have a few conclusions. I'm a little bit ahead of time, but I want to make sure I had plenty of time for questions. So there have been significant progress in drug development in limb-girdle in the past 5 years. We do need patience as these early therapy trials begin. Like I said, we've successfully treated many thousands of mice, but those are not humans. You know, if something bad happens to a mice, we feel sad and sorry about it, but, you know, it's important that we work out the safety and dosing and stuff in humans very, very slowly. So I think, you know, even though there's a lot of excitement and energy in the field right now, we do need patience, and we need to recognize that these early clinical trials will probably treat a very small number of people, and that, you know, both to thank those people who are able to participate in the clinical trials, but also understand that, you know, the goal is, of course, to speed the therapy as quickly as possible so that we can get it approved and treat a broader population of individuals. We are working very hard to develop those tools to reduce the variability that we see in the clinic in limb-girdle muscular dystrophy. That's a very important part of being able to successfully conduct clinical trials. And we're just at the very beginning stages of that, and so we very much need your help because this requires a partnership between individuals with limb-girdle, the researchers, and pharmaceutical companies to develop these therapies. And with that, so I think there's the list of the GRASP LGMD investigators and where they're at, and of course the support from the MDA and Coalition to Cure Calpainopathy for some of the work that I showed you early on. And with that, I'll take questions. Yeah, so I think you had a couple of very good points there. So one of the things that we've done, so there's a couple of barriers when we start to think about how to understand how a form of limb-girdle progresses. Number one, these are all pretty rare conditions, and so to really understand how the disease progresses, you need enough people with a particular subtype to be able and follow them for long enough time. And so one of the commitments that all of the investigators that you've seen here on this list or their institutions have made is to try and share as much of the information that's been previously collected as part of the consortium, to the extent able, depending on how that study was supported. And so I think, you know, whoever you interacted with, we would certainly try to get your prior clinical information from the last study you participated in, and use that as much as we're able to. You should definitely talk to them out back. So I think the 2A is one of the ones that we'll be doing here in Richmond, along with nationwide. So you can certainly stay here, but also Columbus is a great city. So. Yeah, so one of your questions has to do with the early phase gene replacement therapies for 2D, and so we certainly can't speak for what the company will do and how they will pick which clinical sites that they use. From all of the data that's been presented around that study, most recently at the MDA meeting in Orlando, there's certainly a suggestion that trial is moving along efficiently, and of course we all would hope that they would be expanding the sites for that. So, yeah.
I have a question. When you talk about staining to try the muscles and the biopsies and things, when you were staining to try to help determine, can that particular slide be restained and restained, or how much of the biopsy is necessary for you to be able to determine we need to stain in these different
Yeah, so that's a good question. So really what the question is, you had a muscle biopsy at some point. It was maybe showed some stuff, maybe didn't, but wasn't definitive, didn't give a definitive diagnosis. And so when a muscle biopsy is taken, there's nearly always frozen tissue that is retained wherever it was housed. So it could be here in Richmond, it could be in Los Angeles, wherever the muscle biopsy was. That clinical site should retain that muscle, and part of the process from that study is that we would reach out to wherever it was done and obtain, with your consent, a piece of the frozen muscle to be able to restain, again, depending on what was needed to be done. So the short answer is yes. Often there's frozen tissue, and oftentimes we can go back and reanalyze that if we need to.
Is there any retaining time?
What's that?
Is there any retaining time on the biopsy, which taken some time back?
Oh, well, that is very different depending on where. I'm trying to think. Most institutions will retain those muscle biopsies for a very long time. But again, it's very different depending on where you are and what the pathology lab was. And so the only thing you can do is to try. So we can always ask.
Hi there. So both of our boys were diagnosed with 2I, and we see Dr. Catherine Wagner in Baltimore. We saw her a couple weeks ago. She had mentioned that my 13-year-old would be eligible for one of the gene therapies, like trials coming up. Why 13? Why? I didn't think to ask her at the time, but why is 13 that time?
Yeah, so I think a lot of the work that I showed there all was in mice, and so it would be premature on any one of those diseases or molecules to say that we know for sure exactly what age they would use for a particular clinical trial, for example. Part of the natural history study that's ongoing at our site and the others is to help us understand what age is best suited to see a change in that disorder. So I'll give you an example. For example, going back to Duchenne, if you started to look too early on some of those early outcome measures, you could actually see gains in function early on in development. And so that's a good thing for your children, but it's not a very good thing for us to be able to show that a drug did better. So, and so I think a lot of the discussion about what age is best suited for these different clinical trials comes down to safety. So thinking about what age is the right age to start in terms of a safe clinical trial, and then also starting to think about what age where you would be able to see changes during the course of that trial. So there's no guarantee about any age from any of the studies so far. You can keep going. Yeah.
One quick follow-up. How long with the gene therapy until you see some improvement?
Oh, well, that's a good question, isn't it? Yeah, I mean, I think that we won't know until we try the study. That's unfortunately the simplest answer. Yeah, if you see those mice, they get better fast. So they're well, you can't. So you can't. Mice live shorter lifespans than we do, so it's hard to convert that specifically. So the very first things you see in most of the mice studies is that the muscle tissue starts to repair, that it starts to come back and look more normal. That their levels of muscle injury, so the CK level drops quickly. Those are often very early things to see. And then, it does often take several weeks to months to be able to show that the mouse has improved strength or endurance, or however you want to do that. But the one thing you have to keep in mind with mice is that they live in a laboratory, and they're often treated very early in life. So, you can think about it that you would be, it's the equivalent of treating your children when they were, a week to 1 year old. So one of the problems with limb-girdle muscular dystrophy is that as we get older, that cycle of muscle destruction occurs, And at some point, the muscle injury causes that scarring that what you saw in some of my slides and some of Dr. Conley's slides. And so the extent to which those muscles are able to repair is a good question. So our expectation in the prior history is that humans take longer to show a change than mice do.
Yes, go ahead.
I have a quick question. Does children who are wheelchair bound be part of this trial, or would you prefer that it does certify?
Yeah, so the question for this side of the room was, what about if you're in a wheelchair, is essentially what that question is. And so that's a tough question to answer. I think, again, it's hard to know without having these clinical trials approved and posted on clinicaltrials.gov to say definitively that this is going to be exactly the way that this looks for this clinical trial. I will tell you that, the goal at the end of the process is to test a drug in as small a number of people so that it can get to market as quick as possible. So in an ideal situation, you want to be able to expose the least number of people to the risk that occurs when you're testing a new drug, and you want to be able to show as simply as possible that the drug works. Because the sooner you do that, the sooner it's available for every patient with that particular disease. So with that in mind, I think a lot of times when we think about testing clinical trials, we do tend to focus first on people who are able to complete some of the walking tests. But again, there's no statement one way or another about how that will come out.
You mentioned that AGP-15 is being a trial in mice now for 1D. Is that a one-time treatment, or is that?
No, so good question. So the question for that one for the 1D, which is still very much in what we call preclinical work, is that is it a one-time treatment? And so I didn't emphasize that enough, but so gene replacement therapies are typically thought of as one-time therapies. Anything that's a small molecule therapy, this would be something that would likely need to be taken at least at some point during the day or several times a day. So you can think about it like taking Tylenol or aspirin or the medicine you take for your blood pressure. These are drugs that have some sort of dosing schedule and shouldn't be thought of as a one-time therapy. There's a question in the far corner over there.
My family has been studied at the center in Dallas, California, for over 40 years. Studied with my grandfather, and now it's been with my son. So I was wondering, is there interconnection with that information that you guys can draw on that info? I mean, I'm talking 40 years of my family history. Is that available to you with that? Or do I have to find it out and make a phone call to see if that data would be available to you?
Yeah, so it gets to kind of the same question, which is that certainly people and different individual groups have been working on limb-girdle for a very long time, and that's certainly one of the goals of the consortium is to break down those barriers for a lot of reasons, both in terms of your own privacy and safety. The only way that information is ever accessible is with your consent, and likely with the consent of whoever it was that was participating in that, but that's usually true.
The contact information that you have for your study, you went by that slide really quick. Will that be on the MDA site?
I don't know if it's on it'll be on clinicaltrials.gov, and there's certainly information out in the foyer there. So.
Yes, very good.
Two quick questions. One is, I'm assuming that if you don't have a specific identification of your subtype, that doing clinical trials, you're not going to be doing any of that. You don't know what my subtype is. Would I even try to do it?
Yeah, so the first question is, well, what if you don't know what your subtype is? And so, all of these therapies or a lot of the therapies that are being developed are really genetically targeted therapies. And so it's contingent essentially on knowing what the subtype is. as I showed at the beginning, one of the gaps that we identified is that people either don't know what their subtype is because the diagnostic approaches are not complete to be able to get to that point, or that they're, as Chelsea had a really nice example of somebody who had one positive gene and one variant, and so for a clinical diagnosis, that's probably enough, but for a research trial, that may not be sufficient. And so members of the group that I spoke about are working with the genetic information to try to improve upon that. And so that's why that particular project is out there, or will be out there, to help with that issue. So.
And I have a separate question, I don't know if it's really appropriate for the whole thing, but I have to have surgery in a couple of weeks. Does the surgeon and anesthesiologist, rotator cuff surgery, need to know things about my limb-girdle to pay attention to when they're going there and surgery?
Yeah, so the second question had to do with, well, what if you're going to have surgery? Does the surgeon need to know you have limb-girdle, and what does that mean? So the very first answer is that you should absolutely talk to whoever is treating you for your limb-girdle and make sure that they, because different subtypes have different issues with heart or lungs. I think in general, the surgeon and anesthesiologist would absolutely want to know that you had limb-girdle and would absolutely want to have a conversation with whoever's treating you for the muscular dystrophy.
Hi everyone, I'm Kate from Sanofi-Genzyme. Thanks for letting us be here. I just, to kind of get at that, one of the programs that Genzyme offers is called the Lantern Project, which is a diagnostic panel that we run for free. It won't cost your insurance anything. And it does 105 genes that are part of the limb-girdle family. And I brought some sheets that give you that information as well as all the testing information that's available, and all of the labs that perform tests, how they do it, what they're testing for. So if you're curious or you don't know what your genetic variance is, it's usually a blood draw or a saliva test, and your doctor can order the kit.
Yep.
So they're up on the MDA table. It says Pompeii on the front, but the labs are on the back. It's just because it's part of the same panel.
Good point. I think there's one more.
If we participate in the MDA clinics, is your data available for study automatically, or is there something else we need to sign, or is it automatically? Did I sign it already?
Yeah, so unfortunately not. So that, part of participating in a clinical research project is that before we do anything with the information, we have to have your consent. So it's not automatically uploaded from your clinical records, and the reason why, so the consent has two parts to it. One is an acknowledgement by you of all the risks and benefits, whatever they are, of doing the study. That consent is generated through our ethical review process. So, that's your assurance that somebody has reviewed this and making sure that the risks and benefits are acceptable. So, unfortunately, it wouldn't necessarily be. I think they'll talk a little bit more about the MDA mover and how that interacts with your clinical team after lunch, I believe. So.
So even natural history information is not, you'd have to participate in that, you've got to overtly sign something else.
Yeah, yes, absolutely, and it definitely depends on the study. The Lantern Project's a good example. So we have a collaboration with Dr. Heddy at PerkinElmer, who's helping with that project, and so those research groups are collaborating and sharing information, de-identified, so it's without your, it's without knowledge that it's you, but we've been working with other registries to try and share as much information as possible to speed this as quickly as we can. So, but still need your consent.
I just wanted to say that at my last clinic appointment, I signed those forms they brought to me.
So, and then the other point about that is to say that there's probably lots of different studies out there. So making sure that, and everyone has their own consent, so.
All right, we can do one or two more questions before lunch.
Oh, sorry. Back.
Quick question. From listening to your report, it appears to me that either type 1 has nowhere near the possibilities of future than type 2, because you talk more about type 2 than type 1. And then secondly, it appears to me that if you are not in a group that is in the age range or the presentation range of when it starts to affect you, you're probably not a candidate for anything, correct? Except a drug maybe years down the road when it gets.
Yeah, I think you have a.
Right.
Yeah, that's definitely, so you've got there's a lot to unpack there. But the first thing to say is that the research is further along for type 2 than for type 1 because as a treating strategy, it's easier. It's still not easy. It's actually pretty amazing that you can do this in the first place. But people have made many more advances in terms of just being able to replace a deficient gene. So there's lots of ideas about how to translate that into the dominant forms of limb-girdle. And so, even though there's 30 plus different kinds of limb-girdle, there's definitely going to be some that move along faster than the others. I will say that all of the programs in limb-girdle have benefited tremendously from all of the work that's been done over the past 10 to 15 years in Duchenne muscular dystrophy. And so, every time we try and do this in one disease, it gets faster in the next one, which is one of Dr. Conley's earlier points. So that's one thing to say, is that I wouldn't lose hope that, we're not going to continue to march through. The second point is that I think that our group is focused on developing the tools and technologies that you would need to be able to test a drug in any group of individuals, so people who are walking or not walking. And I suspect that trial strategies will depend on what that group is. So, for example, if you have a rare form of limb-girdle, it may be more important to cover all of the population versus just focusing on a certain age range. So I think we're still working on it, so.
All right, I want to thank Dr. Johnson. That was great.